Thank you very much for joining us. It's a terrific turnout this morning, this afternoon, this evening, tonight, wherever you are. Nearly 90 people, so that's excellent. And this is the first of the year. We started this WPC Research Spotlight Series last year, where we had 6. And the idea of this is to bring people who've, made fantastic contributions to research and understanding of Parkinson's disease, and have a sort of little conversation and discussion with them about how they got into research and what they've discovered and such like. So it's great that we've got Ron Postuma from Montreal here with us to talk. I need to just thank a few people. Obviously need to thank WPC, Eli and Kathleen for setting all this up. I want to thank you for joining us. I'd also like to particularly thank Supinus, who are the Supinus Pharmaceuticals, who've actually supported this. Without further ado, as I say, I want to welcome Ron. Ron is probably very familiar to many of you because he's been very pioneering in his research into Parkinson's disease, but Ron's taken it a stage further and actually gone before we even know that people have Parkinson's. He's taken it right back to whether we can track down to when the first problems begin and what they look like, particularly around this particular sleep disorder. But before we get into the sort of research side of things, Ron, I'm always very interested to know how people ended up in research. So it'd be very helpful to hear where you began, whether you began life in Montreal and you've just circled around Montreal, or whether you've been elsewhere, and how you ended up in research in the first place.
Yeah, that's so let's see. I'm from Winnipeg originally. So Winnipeg is a city in the center of Canada. It's actually extremely cold, has a lot of snow in the middle of the prairies. And I fairly knew early on I was going to do medicine, I think. And I've always been attracted, I think, originally to things related to aging, whatever that might be. Conditions related to aging. And so I was going into internal medicine, which is a field of general medicine, I guess, often leads to a subspecialty. And then I was started to become more and more interested in neurology in general and understanding the brain. I've always been sort of interested in that, but it started increasing a little bit. And then, so I decided that I was going to go for 2 years to Australia to do Alzheimer's research. That's the first experience I had with research. So a part of it was being from Winnipeg, which is very cold, and going to Australia, so I did 2.5 years of Alzheimer's disease research then, which is really basic science, like, pipetting and growing neurons and plates and stuff like that.
When did you do that in Australia? Was that in Melbourne?
Yes, it was in Melbourne. Yeah. So I was working with David Small there. It was in the space. Colin Masters, who was one of the people that discovered amyloid, was there as well. So I was at this point halfway becoming a resident or registrar, sort of halfway through that process, left Winnipeg and then did that. Yeah. Okay. And then based on that, I thought, well, neurology sounds good since I'm doing neurology research. And then I managed to get transferred to the Montreal Neurological Institute residency program at the time. And Montreal Neurological Institute is an excellent place to do work. And so they had spaces for residents to go. So I transferred from Winnipeg to Australia, back to Montreal. And I did neurology there, but I retained my interest in aging. I always wanted to work in things related to aging, but then there sort of became a branch. So do I want to stay in Alzheimer's disease or move to something else? And one of the difficulties, and I think I'm glad I did it, is that Alzheimer's disease is fascinating biologically, but there's still nothing much you can do for people. And I am sort of a practical sort of bent. I like to sort of, have positive things that I can do. So I said, well, Parkinson's is like Alzheimer's and it's incredibly treatable and kind of complex. So if you do a good job of Parkinson's, you can make a real difference in everyone's lives. So I moved into Parkinson's. And from Parkinson's, then I started a clinical research career and everything related to Parkinson's.
So why did you give up on the dishes? I mean, you might want to study Parkinson's as opposed to Alzheimer's disease, but growing cells in dishes and studying people in sleep laboratories is slightly different. So Was it the attraction of dealing with a clinical problem, or the awfulness of having to deal with lab work that persuaded you to change direction?
More of the first. I mean, I'm still, I'm very much a doctor, and I see patients, basically every day. I'm seeing somebody with Parkinson's or early stages of Parkinson's, and so if that's what I do all day, then that's what I should leverage, right? That's my skill set. So if I wanted to do basic science research, I have some advantages understanding the disease from the patient perspective, but it's not as direct of a connection. And so there's many brilliant scientists out there who can do bench work after having met a couple of patients, right? But the sort of work that requires working with people really does require someone who knows the disease intimately and thoroughly. And so it makes more sense to work in an area that's clinical research, where you're dealing with people and your expertise goes back and forth. My research helps my clinic helps my research, and so it feels all of one.
Okay. And so when you began, I mean, were you interested in I mean, obviously, we're going to talk a lot about this REM sleep behavioral disorder and prodromal PD. I mean, what was that where you began, Ronald, or was it that it was? I mean, coming back to what you were saying earlier, was it something in clinic you started noticing that people kept saying, actually, I've had a problem before I ever was diagnosed? Or did you sort of suddenly think, actually, I'm just going to take this in a completely different way? I mean, how did you end up with this idea of studying the area you've ended up in, I suppose?
So it's a little bit of serendipity. So I was very much unfocused at the time. I was working in Toronto at the time, and Tony Lang, who is an amazing clinician researcher and the mentor, was I was working on 7 or 8 different projects. I always recommend that when you get started in research, you put your little fingers out. You're like an amoeba. You send out little feelers here and feelers here, and then you have to know which one grabs and which one gets you interested. So I had lots of things going on at the time. I always knew I was going to go back to Montreal because my fiance, now my wife, we had met in Montreal, and so that was always the plan. And we have these journal clubs, and journal clubs are basically where you look at a paper, a newly published paper, and people sit around in the group. There's usually 2 or 3, and we go through them and we discuss them and we think of ideas. And so we looked at a paper by a sleep researcher in Montreal who's a psychiatrist named Jacques Bonplaisir, who's a very famous, well-established sleep researcher, originally a psychiatrist, as I mentioned. And he was talking about treatments of REM sleep behavior disorder, this condition, and it's associated with Parkinson's. And we didn't know very much about it. This was, I guess almost 20 years ago now. But there was a mention in there that this is a very common early sign of Parkinson's disease. And so we all sat around and went, well, Ron's going back to Montreal. He's looking for things that are interesting to do. This is a psychiatrist, a sleep specialist. You're a Parkinson's specialist. Maybe you should go and see these people in the Sacré-Cœur Hospital, a totally different university than I was working, and maybe start a collaboration. And so that's how it all got started.
So it all got started with a journal club reading a paper and thinking, this looks very interesting, and it's a sort of new story, and where's it going to take me?
Yeah, it's a real, the chance favors the prepared mind, but also there's some chance, if you're always got your feelers out, you find the thing that grabs. And so it felt a little bit like that right away. Right when we thought about the idea, we thought, oh yeah, this is going to be an important thing.
Yeah, and I think it's also a very useful message you send for those who are who are on the call who might be junior researchers, that cast that net quite wide when you're starting, but go with what you think you really like to do. Because
exactly,
coming back to your point, there are lots of people who work in the labs who don't really like the lab, and actually, if you're a clinician, it's the clinical interactions that drive you every day, and you've got to do this for many years. So it's great that you have that great desire to sort of help the patients and learn from them, which has obviously been, where the research has gone. So if we go into, I mean, I'm sure most people on this call know what REM sleep behavioral disorder is, but it might be quite a useful starting point because it's going to come up quite a bit. I mean, I often say you're sort of acting out your dreams. And normally you can't because you're actually paralyzed when you're asleep during that stage. But you're the expert on it, Rod. So how would you describe a REM sleep behavior disorder? Because Because if you sort of say, well, you've got a problem with sleep, lots of us will start thinking, well, actually, I've got a bit of a problem with sleep. Have I got REM sleep behavior disorder?
Right. So, yeah. So those of you who don't have Parkinson's, those who, or those, or if you remember back, every once in a while, do you ever get the dream where you're trying to run or you're trying to scream and you just seem like there's something wrong, like you're not running as fast as you should, or you're not yelling as loud as you should? In fact, that's your brain is feeding back. You are dreaming that you are running and you're yelling, and you're actually incapable of running and yelling. That's the normal state. And you can imagine why that would be so important. You don't want people acting out their dreams at night if they're sleeping on the open plain, get eaten by a lion or something while you're defenseless. So, we've evolved this system to paralyze us when we dream. And what goes wrong is when you have REM sleep behavior disorder, this paralysis system stops working. And it usually stops working because of Parkinson's and related conditions, in fact. So it gets old, it doesn't work. And so whatever you're able to dream, you're able to do. Now, it's important to realize it's not the only thing that people do in the middle of the night. So by far more common than that is sleepwalking and sleep talking, which actually happen in other stages of sleep. And so this is especially children and adolescents. Probably about a third of the population has done this one time or another, where you just scream yourself awake, or you see something at the foot of the bed and you scream yourself awake, or you, of course, get up and walk. And there are people who get up and in this semi-half awake, half asleep stage, they drive cars even. I mean, it's really quite impressive what people can do in this other stage. That's not it, okay? So those of you who are freaking out when you hear the rest of the story, you probably don't have that. You probably have sleepwalking, but this is lying in bed, whatever you're dreaming is what you do. And so the people who notice this usually are the bed partners. Yeah. Harold starts having conversations with people, and it really does sound like a conversation. It's like if I was picking up the phone and you were listening to me, and you're in the room next to me while I talk on the phone, and you can hear this sort of, okay, thank you, fine, really, okay, that's what I meant, this sort of thing. You only hear one half of the conversation because the other half, of course, is the other character in the dream. That's a very striking feature of this condition.
And so that would be, so obviously you say there are different types of problems you can have during sleep, of which you've described some, but this sort of REM sleep behavioral disorder we'll come on to discuss its relationship with Parkinson's and related disorders, but that's a fairly firm relationship. Or can people have RBD, REM sleep behavior disorder, and it never moves on to anything else? Can you get it early in life, for example, and it's just a sort of benign condition, or you can get it late in life? It doesn't necessarily mean you'll get Parkinson's or related, but so how much is it associated, I suppose, is the question, really. Right.
So it turns out that about, I want to say sort of 80% of people who really have this problem, not in other words, just think they have the problem, but you go into a sleep lab and you diagnose it and you see it, about 80% of those people actually have it because of Parkinson's or dementia with Lewy bodies or multiple system atrophy, these groups of diseases called synucleinopathy. Okay. I do think there is another 20%. If you ever watch movies about veterans who come back from the war, you will often see the character, after he comes back from the war, is screaming in the middle of his dreams. And post-traumatic stress disorder or anxiety disorders, even antidepressants can actually trigger this sometimes. I think there are some people who normally don't have as strong of a REM atonia system, and if your dreams are horrible enough, if you're anxious enough or aroused enough, you can still act out your dreams. So we do see this once in a while, and there's a few cases, they come in, they're 30 years old, on an antidepressant, maybe a PTSD diagnosis, maybe just an anxious person, often a bit of a family history as well of people in the family doing a similar thing. And when I see those, I think, okay, this is something different. Most of the time, though, it's Parkinson's.
Okay. And when you get the story, I mean, because a lot of people, don't necessarily have access to sleep laboratories, and sleep clinics and such like. Is the story persuasive enough to say, I mean, it's nice to do a sleep study to show that they've got this REM sleep behavior disorder, because obviously to look at rapid eye movements and everything else, you're going to have to do something like that. Or is it actually the story is so characteristic that really you don't need to do these more sophisticated tests? I mean, what would be your view on that?
So it depends on the situation a little bit. let's say someone in front of me has Parkinson's and they tell me the story and it's exactly what I've heard, 500 times, acting out the dreams, et cetera, et cetera. If you have Parkinson's, you got about a 50-50 chance of getting this condition anyway. And so if the story sounds right, that's good enough. That's basically what you have. If, on the other hand, you're someone off the street, okay, and you're a 55-year-old person who has no other health conditions and starts doing this, there are other explanations that could be present. And so let me just close this. I'm getting a pop up here. So, there are other explanations. And even if that would only account for 20%, only about 1% of the population has this condition. And so just by stats, even a test that's say 90% specific, if it's applied to a person who probably doesn't have the condition to start with, say only 1% have it, you're looking at a 10% chance that they actually do have the condition. So in those scenarios, you do need it. So this is a bit of math, but if you have a 50-50 chance without knowing anything else, in other words, you got Parkinson's, you have a 50-50 chance of having the condition, and you got what's called a 90% specific test, that's a 90% chance that you really have it. If you, however, you only have a 1% chance, there's only a 10% chance that you really have it. So it makes a big difference what scenario you're in.
Okay. So basically, sort of summarizing, if you see someone with a sort of typical story in the context of Pugsies, you think that will do. I don't really need to do anything more with that. But when it comes as a sort of isolated phenomena, you have to look at it with more rigor. And I suppose that then takes me into this area of this idea of prodromal Pugsies. So I sometimes get a bit lost myself between what people mean by prodromal Parkinson's disease, premotor Parkinson's disease, and all these different stages, which clearly seem to precede Parkinson's disease. So I suppose it'd be helpful, given your work and what you've contributed in this area, what do we really mean by prodromal Parkinson's disease? Because you've already said that RBD doesn't necessarily give you Pugsies, it gives you disorders which are related. You can get Pugsies, but multiple system atrophy and dementia with Lewy body disease, other diseases associated with this protein, as you say, alpha-synuclein. So when you're talking about prodromal Pugsies, what do you think that actually, what should we be bringing into that rubric, really?
Yeah, so there's a lot of terms and we're really all trying to describe the same thing, and everyone struggles with what's the best name for a condition. Should we call it premotor? Well, what about the motor features of premotor? Should we call it pre-diagnostic? Well, is that just, we haven't got a diagnosis yet? You could come up with a whole lot of difficulties with coming up with a name. So I was involved in the Movement Disorder Society, which is sort of the main body that academic body associated with Parkinson's disease. And we had a task force and we really tried to come up with names for the stages. So let me walk you through that a bit, okay? So first of all, you can be born with, say, there aren't a lot of genes that cause Parkinson's 100%, but say you could be born with a gene and have nothing else wrong with you, okay? That would be considered to be an at-risk state. So there's nothing really going on, but you're just at high risk of Parkinson's. So that's not even Parkinson's yet. But so now, however, the process has started. Some neurodegeneration has happened. The cells aren't working very well. There will be a very, very early stage, we call it preclinical, in which you have absolutely no symptoms, absolutely no signs, but the disease is in your brain and it's starting to do its nasty work. Okay, that's a preclinical stage. At the other end would be clinical Parkinson's disease. And so what that means is a doctor tells you you have Parkinson's because you have slowness of movement with tremor or rigidity, what we call Parkinson's, or dementia with Lewy bodies, which is really very much the same condition with a bit of nuance, or not the same, but very overlapping, where you have memory and cognitive troubles caused by the similar sort of process. So that's clinical disease. So between this preclinical and the clinical is a very, very long period, and we're calling that prodromal. And so what that means is that there is some sign or symptom, the disease is there enough that it's causing something, but you're not at the threshold where a doctor makes a diagnosis. And it turns out that in Parkinson's disease, this prodromal phase is actually extremely long. And so it can range, in some people, of course, it can be very short. They show up immediately with a tremor and nothing else. But in many people, it can be 10, 15, 20 years. And I would say on average, it's over 10 years. So by the time you show up in a doctor's office and the doctor says Parkinson's, that disease has been in your brain for 10, 15, or 20 years on average.
Okay.
That's
So preclinical would be, you could only pick up preclinical with scans and blood tests if we had the sophisticated test by which to do that, because there's nothing to be found either on the history by asking the patient or on examination. And then you start to move into a phase where you, if I've understood this correctly, you don't have the obvious motor features, but things aren't also quite right. So this is where you start to get RBD. And whilst we're on this, Ron, I've had two questions here about, antidepressants. How long after antidepressants discontinued would you be happy to say that the antidepressants cause the REM sleep disorder? And someone else has asked about whimpering during a nightmare with nothing else. Is that RBD?
Okay, so we'll back up there. We'll get back to prodromal in a second. So antidepressants basically can trigger a REM sleep behavior disorder that would otherwise probably be there in the most cases. So it's sort of like a stress test for the sleep system. So most of the time, in fact, when an antidepressant triggers a REM sleep behavior disorder, it actually is real REM sleep behavior disorder under it, and the antidepressant made it showed up earlier. Okay. Whimpering, little things during nightmares, little normal. That very many people do that, and I don't think it means very much by itself. Usually we're talking about pretty obvious things when we're talking about REM sleep behavior disorder.
Okay, so thank you very much for answering those two questions. So as we move into prodromal PD, prodromal Parkinson's, obviously we have this REM sleep behavior disorder, and as we described, you have to define it and understand what it means, and also not confuse it with other sleep disorders. What else would you like to see within that, if you like, within that sort of phase, which makes you more confident that this is a Parkinsonian prodrome?
Yeah, it turns out that, in fact, this prodromal stage is all over the place, and there are many symptoms that you can get. By far the most common symptom would be olfactory loss, and that's the earliest, and we think on average that can start 20 years before the disease, so you can't smell as well as you used to, and many people are unaware of it because it creeps up on you gradually, and you don't really notice if there's no smells. You just think there's nothing to smell, right? Constipation is another one, and that one's so early you wonder if it's also a risk factor as well, and then there's a variety of other what we call autonomic things. So dropping in blood pressure when you stand up, having to pee more often, getting some erectile dysfunction. All of these are very common in people who don't have Parkinson's, but they're actually a feature of Parkinson's. And the reason for these other things, those two, is that the disease is not even starting in the motor areas. So the disease is starting, we think, in the olfactory areas, okay, and probably also at around the same time in the areas that control this autonomic nervous system, including way out in your skin, in your colon, in your salivary glands. The disease is actually starting out there at around the same time that it's starting in the brain as well. And so this is really fascinating because, and very important clinically, because it means that there's this disease process that's potentially kind of walking around your brain and walking around your body, moving maybe even from cell to cell, and it does so over 15 and 20 years. And maybe that's a really huge window to do something about that, if we could ever do something about that.
Okay. Yeah, so that'd be good to come back to that, because how we can actually, apart from just being useful in terms of being more confident that that's where the patient's going, if you like, into the condition, what that actually means therapeutically. So, but I suppose what you're saying is, obviously losing a sense of smell can be due to all sorts of other reasons. So it's a sort of combination of this, which is often the clue, really. I've got a bit of REM sleep behavior disorder. I'm a bit more constipated. My smell's not quite so good. And it's that sort of everything together isn't quite right. None of it's bad in itself, but together that's what gives you the sort of prodrome, if you like.
That's right. And so, in fact, we have these diagnostic criteria for the prodromal disease, and it's actually a mathematical calculation where you sort of add up all of these based on how diagnostically strong they are. So constipation, I mean, almost everyone is constipated at some point. This is not a very diagnostically strong marker. Olfaction is pretty good, actually, but olfaction, say, increases your risk of 5 times. Well, that's a lot, 5 times, but still, your lifetime risk of getting Parkinson's is 1 in 50. So now maybe it's only 1 in 10. That's still 9 out of 10 chance you got something else causing your olfactory loss, for example. Yeah. But when you start putting these together, you can start getting pretty high confidence that a person has it. The only diagnostic marker that's strong enough basically on its own in many people to make a diagnosis of prodromal Parkinson's is REM-seq behavior disorder. Everything else is non-specific, but REM-seq behavior disorder, like I said, 80% of the time, it's actually Parkinson's.
Okay, so now, I mean, there's two questions related to that, I suppose. One is we had Per Borghammer on last time, who has this, view that the disease either begins in the gut and goes centrally, or stays in the brain and then goes peripherally. And so, his argument would be that's why sometimes you see RBD prodromal and sometimes you don't. And I suppose one of the questions would be, how much do you buy into that idea that there are different types of disease starting in different places? And I suppose linked to that will be, how do we understand how RBD relates to different types of synucleinopathy? So why do some people with RBD end up with Parkinson's, some with dementia with Lewy bodies? And why do some people with RBD get Parkinson's next year and some people wait 15 years to declare themselves?
Yes, right. So
Simple questions.
Yeah, there's a couple of nuances to that. So the idea, first of all, it is true that patients with Parkinson's, not only half of them have RBD, right? And if you have RBD, it's usually less of a good sign, actually. I think there's various ways that we can look at it. So one way of understanding it would be that there's a different pattern of spread, like Paris says, top down versus bottom up. Absolutely possible. Another explanation that could also be could actually be not mutually exclusive, could be part of the same thing. But, we've often thought about Parkinson's as being pure motor or relatively diffuse. And so if you see the synuclein going in all areas of the brain, they get REM-seq behavior disorder, their blood pressure drops when they stand up, their cognition is not so great, they got a lot of walking dysfunction maybe, as opposed to this pure, perfect motor person, that's it, maybe a bit of constipation, their sense of smell is lost, but really it's a tremor, and they have fluctuations and dyskinesia. There's a spectrum there. And you would obviously rather be on the pure motor spectrum, because as frustrating as the fluctuations in dyskinesia are, there's a lot of other problems, that diffuse thing that you don't have. And so I think there's a distinction, that there's definitely a different pattern of neurodegeneration. I think it might go top down, bottom up. It might just be, pure, you present early with Parkinson's only, or you don't present with Parkinson's right away, and by that time the disease is all over the place by the time you show up in the doctors. So pure versus diffuse, these are all various ways of thinking about it, and I don't know what the right answer is on that one.
And if you had to sort of pinpoint the pathology underlying RBD, I mean, so obviously in the bottom of the brain, the brainstem at the top is where the dopamine cells are, and that's what obviously most people associate with pathology. So where's the critical pathology in RBD in the brain?
So it's probably in one of two areas, probably two, in fact. The most common one talked about is in the area in the pons. It's just below the midbrain, and it's called around the locus ceruleus, the perilocus ceruleus area, and the locus ceruleus. And this is involved in transmission of noradrenergic, but even the next door area is probably a flip-flop switch that flips you in and out of paralysis during sleep. And so we think that that's the primary area that goes. And that has a connection a little bit lower as well with a nucleus in the medulla, gigantocellular nucleus in the medulla. So there's a bit of a circuit, in fact, that might be affected. So is this the one that causes it, or is this the one that causes it, or is it both? We don't exactly know in the majority of cases. I think most of the time you're seeing a pretty broad brainstem degeneration, so it's often impossible to say exactly what nucleus is involved. But yes, it's down in the brainstem area of the brain, down a little bit lower than the motor areas, and usually earlier in the disease process.
And you're saying if you do have RBD and develop Parkinson's, or you get RBD with Parkinson's, that tends to be slightly less good?
Yes, yeah, it's this. It's almost I call them the four horsemen of the apocalypse sometimes, and it's one of the four. And it's really bad autonomic dysfunction, especially a drop in blood pressure when you stand up, cognitive issues, a lot of gait dysfunction, and REM-seq behavior disorder. You see those, they tend to run together. They're very hard to pull apart. Patients tend to have 3 or 4 of those, and is either 0 or 3, of these things. Yeah. And so when you start to see those things in combination, this is what we've been calling this diffuse malignant subtype of Parkinson's disease, where it's, things are clearly worse on average. Okay, some people do great. than the pure motors, or this might be the body up if you want to use a different version. But there is some prognostic significance to REM sleep behavior disorder for sure. It's not absolute though.
And I mean, this is unknown, I think, but I suppose the question would be, is that because the disease has spread and sort of hit critical nodes in the brainstem, and it's a sort of marker of a more aggressive disease, or do they feed off each other? So there is this idea that if you keep dropping your blood pressure, you keep underperfusing your brain, and that doesn't help your thinking. And obviously there's this whole area now of sleep hygiene and the value of sleep in clearing away proteins and things that are bad for your brain. So I suppose the question is the extent to which they feed off each other. So if your blood pressure keeps dropping, that affects your cognition. You don't sleep properly, so therefore you can't get rid of synuclein. So the processes actually are driving the disease.
Yeah, I suspect it's not that, but I don't know that to be the case. I think us humans are very good at making causal chains, and we're making causal chains all the time. But in fact, if you wanted to argue the opposite, you could probably come up with a pretty good causal chain for the opposite too. I think this is probably the REM sleep behavior disorder, for example. I don't think it's causing a lot of damage per se. Yeah. It's a sign. It's an indication that the synuclein is here.
So if you treat the RBD, so if you treat it with clonazepam or whatever, it doesn't change, because you say it's associated with, the four horsemen of the apocalypse. It's one of the bad signs to get. But if you target RBD and say, well, I can do something about that, some of these other problems are harder, it doesn't actually change what happens thereafter.
It does not seem to be. Yeah. In fact, when we've tried to look at this, we look at people who are treated, and in fact, on average, the people who are treated do a little bit worse than the people who decide not to be treated. Now, I don't think that's causation either. I think it's if you have more severe symptoms, you're more likely to be on treatment. But we see no sign. I've seen no sign that treating RBD makes any real difference. It doesn't really affect the rest of your sleep very much. The rest of your stages are pretty good. Even your REM sleep most of the time is pretty good. It's just every once in a while you wake yourself up because you knock the bedpost or something like this. The sleep in RBD patients is generally all right. It's more of a sign.
Okay, and then coming back to this issue of I suppose it's difficult to come up with a clear answer to this, but because the implication for what you're saying is RBD is indicative of a more aggressive disease course, and yet we also have people who have RBD from your own studies going 15 years before they declare themselves. So I guess it does cover a spectrum here, that RBD, and I suppose this is what some of the questions which are coming in, RBD as a population of people with Parkinson's disease are bad, or carries a worse prognosis, but on an individual basis, it's harder to be certain on that.
Yeah, I don't want to oversell this. Half the people have RBD, and half of my patients with RBD are just fine, and there's nothing wrong with them. It's a risk factor, and that's all it is. Okay. Actually, I'm circling back a little bit. You mentioned that they don't just get Parkinson's, right? They also get dementia with Lewy bodies, and they also get something called multiple system atrophy. But it really is related to this protein called synuclein. And the question about why some people get dementia with Lewy bodies and why some people get Parkinson's when they have the same protein is a really fascinating one. And I think there's lots of answers to that. We shouldn't forget that about 25% of us at the age of 80 have early changes of Alzheimer's in our brain. And dementia with Lewy bodies is what happens when you have Parkinson's and Alzheimer's at the same time. So if you have REM sleep behavior disorder, you got the synuclein starting to march around your brain, and it hits an area where early never would have been a problem, Alzheimer's is present, those two things together could be a nightmare. And so maybe that's the person who develops dementia, for example, as opposed to Parkinson's, or maybe it's the protein is different and spreads in different ways. We don't really have a good sense of why that's the case.
Okay, and so coming back to something we were discussing earlier, I mean, obviously this is fascinating insight into what goes wrong with synucleinopathies and Parkinson's disease, and this gives us a sort of window into seeing some of the earliest pathological changes and the consequences of that. I suppose one of the questions is, is that a useful thing to know? So if I come to you, Ron, I've got a REM sleep behavior disorder, you're going to say, well, Roger, you've got an 80% chance of developing Parkinson's or something like that in 15 years. And I'll say, that's great. What are you going to do about it? And you'll say, I'll just follow you up.
Yeah, yes, I say go out and get exercise. It is somewhat discouraging. Exercise probably makes a difference, but I can't say that for sure. Yeah, so this is where we need to get to the next stage. I mean, The reason I got into this almost 20 years ago was to do something practical about it. It's obvious to everybody. I mean, this is so crashingly obvious, is that if you could sort of try to bring these drugs in that we think might slow down the progression of disease, it would be so obviously better to bring them early before a person ever gets clinical Parkinson's or dementia. Everybody knows this, right? The difficulty now is translating that thing that everyone knows into actually doing it. Yeah. One of the difficulties is that we don't really have a preventative agent for Parkinson's. And I think you could come up with many explanations for that. And I think one of the compelling ones is, in fact, by the time you have Parkinson's disease, the disease process is pretty well established, and we might have already missed the chance to make the real differences in some of the symptoms. That's one argument, I think, which has to be paid attention to. The other argument is we just don't have the drugs that are good enough yet. You know, everything helps a little bit, but nothing is good enough. But there are definitely some medications in the pipeline now that everyone is kind of excited about. And most of the ones are either focused on the genetics of Parkinson's disease or are focused on the synuclein. And that's what gets most people very excited. Inflammation maybe being another one. Yeah. Now the next step is take these medications that we know should be used early and sort of convince the companies who make $100 million bets, or maybe government agencies who could have access to large-scale funding on these, to try these in earlier stages of disease where we know a little bit less about how they evolve, how the disease changes over time, how many people do you need to do a study, and all the very nuts and bolts of what's the chance of this agent, if it works, what's the chance of a successful trial, if assuming an agent works. And we have a better idea about that in Parkinson's, I would say, although we failed maybe because it's too late. We have less of a clear idea of what would happen if we started doing these trials. And so it really just takes someone to take that next step and start putting these drugs, testing these medications in people in these early stages. And we're ready to go. I mean, we'll start.
But has anyone done that? So are there any trials of so-called disease modification in patients with RBD?
No. We have about now, I would say, the collected centers of the REMS Behavior Disorder Study Group, which is a group of people who are really all interested in this. We could probably get 5,000 patients mobilized, potential candidates mobilized. It just takes someone to say, okay, here's your $20 million, which is what these trials cost. They all cost this. Let's go and let's try it, and we'll go.
And I guess, I mean, I've only just thought of this, but I mean, this may be a stupid idea, but I'll throw it out there anyway. I mean, if you've got disease-modifying therapies in this stage of the disease, one of the arguments against it has always been, well, if it takes up to 15 years, how do I know I've had disease modification when it's so variable in terms of how long it takes to get to a clinical state that I recognize, and how long, and also those are variable states. But I suppose the other way of looking at it, if you stop someone having RBD with a disease-modifying therapy, would that in itself tell you you've modified the disease? Because if you're saying that the RBD is due to synuclein or cell pathology, if you got rid of RBD without them having drugs, surely that's an evidence for disease modification.
Maybe. The only thing is that the symptoms of REMS behavior disorder are incredibly variable. So, if you're having a rough time, you're stressed out about things, your dreams get more intense, your dream enactment behavior goes way up for this exact same amount of pathology. And so theoretically, we could measure the changes in that nucleus. So it's 60% gone versus 55% gone versus 50% gone. We could do it that way. But when we've tried to look at whether
so this is the locus ceruleus, or this
is
the,
yes, exactly. Or
we're doing it with PET or MRI, or
I would be talking here about, say, a sleep study where you measure how much the tone system is gone, for example. Okay. You can do it, and it does progress with time. But if you actually practically, it's just not a very good marker. But I do think, we don't have to do 15-year trials here. We've been working for decades now on finding all the other predictors of disease. And so now you have REM sleep behavior disorder, but not only do you have REM sleep behavior disorder, your sense of smell is gone. And we have a test of motor speed, and you're in the bottom third of the population on that. Okay, well, we're not talking about a 6% chance of getting Parkinson's in a year. We're talking about a 15 to 20 to 30% chance of getting Parkinson's in a year. Well, let's put you in the trial, okay? A little bit closer to disease. We've lost a little bit of a window to intervene, but now we can start doing trials that are 2 or 3 years long, which is the standard length of time for a pharmaceutical trial. No one can do a 15-year trial. It just isn't going to work. You know, the patients will drop out. The practicality is it's insane, but everyone can do a 2 to 3 year trial. And so I think we're ready to go with 2 to 3 year trials now.
So you would see that the sort of future is that we would probably be better off investing these. So someone's talked about alpha-synuclein antibody trials and all these disease modifying trials, but actually going for people who've been stratified to be at a high risk of developing a synucleinopathy, of which Pog's is the commonest one, within the next 5 years, enrich for that population and then treat. And then ultimately, if that is successful in some way, you could then roll the clock back, because obviously your point at the beginning was the earlier you go, the better it is.
Right.
But this strategy obviously moves it towards the end of the prodrome, if you like.
Yeah, so you do it for practical reasons because that's the only way you can get the trial done. Once you have a successful modifying therapy, you know, no problem, you can move earlier, right? I would just take it. If you establish a disease state, I mean, if you're preventing the disease from getting worse from year -3 to year -1, it should also prevent the disease from getting worse from year -13 to -11, right? I mean, it's the same disease. But
and I think also, I mean, one of the points I always try and make in clinic is you know, based on your work, you know, that this disease has a run-in period of 10 or 15 years at least.
Right.
Then I always think it's very helpful for patients, because they realize by the time they present, they've already had condition for 10 or 15 years. So it's not suddenly going to change overnight. It's going to carry on. Yes. And obviously, if the disease course is 25 to 30 years and you slow it down by 50%, that suddenly takes it from 25 to 30 to 50 to 60, and your average age, you get it 60 to 70, someone else will get you. So I suppose, you know, this idea of cures is obviously very attractive, but slowing disease processes down by, you know, 50% would actually be major impact. So based on your studies, what you just said, you know, if you slowed it down by 50%, instead of taking 3 years from a prodromal to get it, it's 6 years.
Yeah.
And then, you know,
and then if you keep taking that drug, you know, instead of say a 15-year disease, you now got a 30-year disease. And it's exactly like you said.
So I'm interested in this sort of this group you've got together who sort of work in this area. I mean, I think it might be helpful for the community to sort of hear how this came about and what its sort of vision is, if you like.
Right. Well, there's a lot of groups that are interested in this. So, for example, I'm involved in a big study with the NIH called the NAP study, where a North American Prodromal Synucleinopathy study. These are very formal, large studies that sort of run big trials, sort of really following patients carefully. This is a little bit different. This is just a bunch of people who do research in the area who get together when there's not COVID and bounce ideas around each other and try to combine forces. So, you know, this is a condition that affects 1% of the population. So that's not rare. That's a lot of people. But who actually shows up to the doctor? A vast minority of those. Most people just think it's normal. And so every center, a sleep center, has 40 here, 50 here, 100 patients in the big ones. This is the kind of the numbers of people that we have available to us. And so we decided that we're going to combine forces together and try as much as possible to share projects. So share data that we collect from patients to really sort of answer the questions about that take 1,000 people or 2,000 people in order to answer. And so that's what we are. Yeah.
And the idea would then be that you've got a network. So when a therapy comes along, it's easier to sort of roll it out across that.
That's right. So, you know, pharmaceutical company A is ready to roll out a prodromal trial, calls up us at the REM-seq Behavior Disorder Study Group, and a mass email goes out, and 5,000 potential patients are in your trial already. And the first pharmaceutical company to do that gets all 5,000, and the next one gets the leftovers. So, you know, there are probably enough patients around the world now to run 2 or 3 or 4 pharmaceutical trials. Of course, the first one to do it has the easiest job. There's an immense amount of pent-up demand in our group. We're all ready to go.
And where is the resistance? Because I mean, your arguments are very persuasive and your data is very persuasive. So I guess the question is, where is the resistance coming to this? Is it because the the companies have yet to buy into this concept? Is it because the FDA don't recognize this as disease modification, so there's no reimbursement, so why do a trial if I'm not going to get it anywhere? I mean, where do you see the sort of blocks in this as a sort of going forward?
Yeah, one of the things is no one's ever done one before, so we don't want to be the one who makes all the mistakes when we do it in the first time like this. And so a lot of it is we have to show them, people that we can de-risk this and can do this, and we know what's going to happen. I think we're pretty good at that now, but if you're coming at it from the outside, you might not see it, like we've been working in the field for 10 years. The FDA has often brought up, or EMA, that there may be no clear licensing path. I personally think that's a false issue. You show that you slow down a disease in its prodromal stages by 50% and reduce the occurrence of a disease by 50%, disseminate that to the general public, and then watch the FDA say no. They will not. If you have convincing science, the FDA are human beings who know what they're doing, and they're generally good people. They will read the literature when it happens. I personally think that that's the case. I have trust in the system, if you will. So I'm not worried about that. But I think it's a lot of just no one quite know. It's reluctance, a bit too much reluctance, I think, just because no one wants to be the first, I guess.
Yeah, I mean, it is strange, isn't it? Because I always remember hearing a talk, I think we were both at the same meeting, and it was in Toronto, where they talked about the 33 amyloid antibody trials in Alzheimer's disease, which were negative. And, I don't work on Alzheimer's disease, but I think one could be fairly confident that was not working as a therapy. And yet people still carry on working at it. And they always say you need to go to earlier stage and such like. And actually, it's harder with Alzheimer's, whereas with your data, it's pretty convincing where you need to go. And it would seem to me that a lot of the arguments for these diseases like Parkinson's, Alzheimer's disease, if you can push the window back of when people first present, so average age, you say get Parkinson's is 70, you move it to 80, like Alzheimer's from 80 to 90, you will have made a huge impact on the costs of these diseases, both personally and to society, by simply shifting it back without getting rid of it. So,
yeah, and Alzheimer's is a great example. We'd be throwing these amyloid therapies. Everyone just did the same thing, and then we started moving earlier. But now we have a hit, right? I mean, we don't know, but there is a positive trial of an amyloid drug, and it's been licensed by the FDA, and it's insanely controversial. And no one really knows whether it really, really makes a difference, but there certainly is some sign. They went to prodromal disease, and that's where you get a licensed product. So I hope that example of Alzheimer's disease will apply to the prodromal state in Parkinson's as well, because it's almost exactly the same thing. Move the drug earlier, and you're more likely to be successful.
So you think if these alpha-synuclein trials, which are obviously ongoing at the moment, and for which we mainly just have safety data rather than any efficacy, if they are negative, they don't work, do you think they'll come knocking on your door?
I don't know. I think they should. If they don't work, there's a compelling reason. It takes a long time to kill a neuron by preventing it from, say, getting infected by synuclein. The process may be slow, and 1-year trials and 2-year trials, may not be enough. You put it out to 3 years, put it early, and it'll work. Too little, too late is the most likely explanation for a failure when we get it. Not that the target's wrong. I think the target is probably correct. We should be targeting synuclein. It's whether we can make a difference quick enough that we can actually see it, and that's the harder question.
So I suppose we're coming towards the end now, but it sort of just, I mean, your work raises many interesting questions about prodromal PD, and then it's sort of there's a sort of idea about screening. So obviously you don't want to take the entire population into a sleep laboratory or sleep clinic, but there are portable devices. Nowadays there are Active watches, there are all sorts of Apple Watch, all sorts of things for monitoring activity, if not EEG activity, brain activity in sleep. So I guess the question is the extent to which we should be now thinking about, are there cleverer ways of diagnosing these sort of conditions in the community? And then how much should we be, we've just talked about the fact we haven't got a disease modifying therapy, but if we imagine that may come along, should we actually be screening the population in general for these type of disorders, and actually, rather than waiting on people to come to us? I mean, what do you think?
Yeah, so Obviously, I think we don't want to go screening until we have a disease modifying therapy, because it really does no one any obvious good to take a person off the street, pull them off the street and tell them they have prodromal Parkinson's, right? But can we do it? Well, we actually tried. I'm not going to do it again because I don't know if I can do it, but we tried once to put a little newspaper ad, and we simply said, do you act out your dreams at night? Basically a question, call us. And then we got about 120 calls or something like that. It was a tiny little circulation magazine. And then my research associate, Amélie Pelletier, who, she's not an MD, although she's unusually smart, just sort of did some screening and said, that sounds like it could be it, and it might not be. 70% of the people that she thought could have RBD, in fact, did have RBD when we did the study. So you can definitely screen for this. If you're aware of it, you can screen for it, no problem at all. Making a diagnosis in the clinic, not recurring a sleep study, that's also that's a separate problem. After you screened, do you have to bring them into a lab, or can you mail them a device? Well, you basically need to know if you're asleep, and you need to know what stage of sleep you're in, and if you need to know whether you're paralyzed. And you can't just know whether you're paralyzed, because you could have just woken up and started moving around. You can't know if you're sleep disrupted, because that doesn't tell you anything. You need those things in combination. So a lead on the brain, a lead in the arm, and you should be able to do it.
Okay. Because I think a lot of people get a bit confused with this. I mean, even I fall into this category. I wear my Fitbit at night, and it tells you how much sleep you've had. So you go away and say, well, I had 8 hours sleep last night. That's terrific. But of course, what it's telling you is you didn't move for 8 hours. If you just lie still in your bed for 8 hours, it will tell you you've got 8 hours. Or if you take it off, it will tell you you've got 8 hours sleep. So I suppose that the point you're making is, if we really want to start looking for this out in the community, we are going to have other devices which directly record brain waves and muscle tone as opposed to movements.
Exactly. We need the two of them together.
Yeah. Okay. Well, that's been very helpful, Ron. We've had lots of interesting questions come down the side about lots in the chat about different types of Parkinson's and such like. I think we've answered most of these questions. I suppose the last thing I would just ask, which seems to relate a bit to the last question, which is the extent to which we can model any of this in the lab. So obviously, this is clinical research, clinical experiments, if you like. How far have we got in the lab in modeling this so we can start screening for agents? We've already talked, there's lots of agents around synuclein and disease modifying therapies, but how well are we at modeling prodromal Parkinson's disease in the lab, and can we use that as a way of finding agents which we could then bring to the clinic?
Well, there are a few groups who've been able to produce REM sleep behavior disorder in mice, for example, and there's a variety of ways to do it. You can overexcite them with the transmitters. You can burn a hole in the area that's involved. Those two approaches would not necessarily tell you about disease-modifying therapy, because they don't recapitulate the process. Another one, injecting synuclein into areas of the brain that are connected with the REM atonia systems, has been tried as well, and that has shown things that look like REM sleep behavior disorder as well, and some early work. That's getting there. That's getting to be a model, I think, that you might be able to use. So there are some basically around the synucleinopathy that I think I might start to trust in terms of disease modification. The other thing, of course, you could do is ignore REM sleep behavior disorder altogether and just model the disease with synuclein injections and growing organoids and cells. You don't necessarily need the RBD itself, because it's a broad prodromal Parkinson state, and so whichever way you can get at it, it's good.
Okay. So basically, animal models, you can model aspects of the disease, you can model aspects of the protein aggregation, but sort of modeling RBD, unless it's sort of really mimics what you see in the clinical population, has limited potential, and actually using any of these models would be helpful, which we already have for looking at disease modification.
Probably for disease modification, or yeah, exactly.
Yeah. Well, sort of our hour is up. I have to say it's been a very stimulating hour. It's been very, very interesting. I mean, it's a sort of conceptual leap of moving our entire resources in Parkinson's disease. And I wouldn't want people on this call who have Parkinson's to think we're abandoning the Parkinson's population for this prodromal. But I think this idea that there's this continuum, as Ron was saying, from preclinical to prodromal to overt Parkinson's disease, seeing that and then trying to move our therapies earlier whilst not ignoring what we can do later, is clearly going to be important. And you've heard a lot about some of the difficulties Ron has had, as others, in persuading how to do this in sort of big trials. But it's been a great to hear you, Ron. Many congratulations on taking on this topic. I mean, of course, what people don't understand is when Ron says 80% of people get a synucleinopathy after 15 years, it means Ron's had to follow people for 15 years. It's not a figure that just appears. So that dedication to actually studying this, understanding it, and then actually what that actually means has been fantastic, I think. And not only in terms of understanding Parkinson's disease and our treatments for the future, but it's actually transformed what you do in the clinic. So I remember very early on, trying to work out whether people have early Parkinson's disease. And so you spend a lot of time, is there a bit stiff, is it not? And now you could just start asking them these questions about, have you been acting out your dreams? And then they say yes, and you think, okay, now we know where we are, rather than this sort of rather subtle clinical features. So thank you very much for all your work. Thank you very much for this very stimulating hour. Thank you, everybody, for the massive questions. I'm sorry if I didn't get to your questions. Some of them are quite long, and I find it very difficult to read them whilst listening to Ron's very interesting answers. But thank you all very much. That will conclude our discussion today. The next one of these research spotlights is on the 24th of March. And we're going in a slightly different area. We're going to go into the genetics of Pug's disease, which Ron mentioned, these genetic forms of Pug's disease. And we've got Andrew Singleton, who's going to be trying to tell me and you whether genetics has anything to tell us usefully about Parkinson's disease. So thank you all very much. And particularly, thank you very much, Ron, for your time with us this afternoon.
Thank you. Take care.